All right, buckle up because today we are diving deep into the world of those tiny but mighty molecular machines, enzymes. You guys gave us a treasure trove of info on these essential workers, and I, for one, am ready to dig in. What do you say we jump right in?
Well, most folks don't often think about enzymes, but they really are the unsung heroes, quietly working in the background to orchestrate the reactions that make life possible.
Kind of like a stage crew making sure the show goes on. Only instead of lights and music, we're talking about the molecules of life itself, right?
Exactly. And they're involved in everything. Think building DNA, helping you digest your lunch, even that fizz you see when you put hydrogen peroxide on a cut. That's an enzyme in action.
They're like specialized tools, each designed for a specific job. So let's start with one we've all heard of, DNA. It's the blueprint of life, but how does it actually get copied? Enter DNA polymerase, right?
Precisely. DNA polymerase is like, imagine a high-speed copy machine. But here's the kicker. It also has a built-in spell checker. It moves along the DNA strand, adding the right building blocks to make a perfect copy. And that spell checker, that's its proofreading ability, fixing any errors as it goes.
So it's like having a super efficient scribe, making sure the blueprint is replicated without a single typo.
Speaking of efficiency, have you heard of Taq polymerase? It's a special kind of DNA polymerase found in bacteria that live in those crazy hot volcanic vents.
Wait, bacteria that live in volcanoes?
It's wild, right? And their enzymes are just as tough. Taq polymerase can withstand insanely high temperatures, which is why scientists love it. Remember PCR polymerase chain reaction? That technique relies on Taq polymerase. It lets scientists make millions of copies of DNA from even a tiny sample. These are for everything from diagnosing diseases to solving crimes.
So we've got this heat-loving bacteria that's helped revolutionize entire fields. Okay, so DNA polymerase is the builder, but what about the messenger? That's where RNA polymerase comes in, right?
Absolutely. If DNA polymerase is the builder, RNA polymerase is the messenger. It transcribes the DNA blueprint into RNA, like going from a master plan to a set of instructions for a specific task.
So it's translating the blueprint into actionable steps.
Exactly. This process is key to gene expression, turning the info locked in DNA into something the cell can actually use.
Makes sense. But I read this process isn't always a smooth ride. Something about abortive initiation.
Ah, yes. It's a bit like starting a car on a cold day. Sometimes it sputters and stalls before it finally turns over. RNA polymerase can stumble at the start, making these short, incomplete RNA transcripts before getting it right.
So it's like a bit of trial and error before sending out the message.
Exactly.
Okay, so we've seen how DNA is the template for RNA. But what happens when you flip the script? That's where reverse transcriptase comes in, and it's a real game changer. It's like rewinding a cassette tape, totally unexpected.
Great analogy. Reverse transcriptase does exactly that. It goes from RNA back to DNA. And it's crucial for retroviruses like HIV. They actually use RNA as their genetic material. So they need reverse transcriptase to insert their genes into the host cell's DNA.
Which is why those reverse transcriptase inhibitor drugs are so important in HIV treatment. They block that crucial step.
You got it. They basically stop the virus from hijacking the cell's machinery to make copies of itself. And, this remarkable enzyme isn't just essential for viruses. Scientists use it in the lab, too. It's a key player in RT-PCR, a powerful tool to measure gene expression.
Talk about a multi-talented enzyme. Okay, next up is an enzyme that sounds like it belongs in a skincare commercial, telomerase. It's like the fountain of youth for our chromosomes, right?
In a way, yeah. Imagine you have those little plastic caps on the ends of your shoelaces to keep them from fraying. Those are like telomeres. They protect the ends of our chromosomes from damage. And telomerase, it's the enzyme that builds and maintains those telomeres.
Okay, that makes way more sense than a fountain of youth. So why are these telomeres so important?
Well, every time a cell divides, those telomeres get a little bit shorter. It's like a cellular clock ticking away. And eventually they get so short that the cell can't divide correctly anymore. That leads to cell death, or the cell just stops working properly.
So telomerase is trying to slow down that clock, at least for our cells.
That's it. It's constantly working to replenish those telomeres. But over time, telomerase activity tends to slow down, and those telomeres, well, they just keep getting shorter.
Fascinating. So how does this all tie into aging then?
Well, as more and more of our cells get to that point of no return, we start to see those classic signs of aging wrinkles, a weaker immune system, and things just don't heal as well as they used to.
It's like our biological machinery is slowly wearing down.
Exactly. And it's not just about how long those telomeres are. It's also about how active telomerase is. And get this enzyme has its own little world inside of it. There's a subunit called TERT, which is actually a type of reverse transcriptase, an enzyme within an enzyme.
Wow, talk about a complex machine.
It really is. And, scientists are still trying to figure out all of its secrets. Like, there's some research that says things like chronic stress and unhealthy habits can actually speed up telomere shortening. But on the flip side, things like exercise and a good diet might help keep those telomeres longer.
So there might be ways we can actually influence this cellular clock.
It's a hot area of research, and while we don't have all the answers yet, it's a fascinating glimpse into how our genes, our lifestyle choices, and aging are all interconnected.
So from replicating DNA to playing a role in aging, enzymes are really the little engines that could. But let's shift gears from the microscopic world of our cells to something that powers life on a much grander scale, photosynthesis. I hear there's an enzyme that's a total rock star in this process, Rubisco, right?
You got it. Rubisco is absolutely essential for photosynthesis. That's how plants use sunlight, water, and carbon dioxide to create sugar, the building blocks for their growth. And since plants are the foundation of most food chains, including ours, you could say that Rubisco is indirectly responsible for sustaining life as we know it.
Wow, that's a pretty big deal for a single enzyme. It's like the ultimate provider.
Absolutely. And it's not just what it does, but how much of it there is. Rubisco is potentially the most abundant enzyme on Earth.
That's a whole lot of carbon dioxide being turned into sugar. But there's got to be a catch, right? No system is perfect. What's the downside of Rubisco?
You're right. There's a trade-off. As amazing as Rubisco is, it can be a bit inefficient. Sometimes it accidentally grabs oxygen instead of carbon dioxide, which leads to something called photorespiration. And unlike photosynthesis, which gives the plant energy, photorespiration actually wastes energy.
So it's like Rubisco has a bit of an identity crisis, sometimes working for the good guys, sometimes accidentally helping the bad guys.
Kind of. Scientists are actually researching ways to make Rubisco more efficient. They hope to boost crop yields and maybe even help fight climate change by enhancing carbon capture by plants.
That would be incredible. Okay, so we've got an enzyme that builds, one that transcribes, one that reverses, one that protects, and now one that feeds the world. That's quite the resume.
And we're just getting started. There are countless other fascinating enzymes, each with their own story to tell and a vital role to play.
Well, we're ready for more. What other fascinating enzymes are we diving into today?
Let's talk about an enzyme that's like a cellular detox crew catalase.
Oh, yeah, right. That one deals with hydrogen peroxide, right? The stuff you put on cuts.
Exactly. Our cells actually produce a little bit of hydrogen peroxide as a byproduct of metabolism, and it can really do some damage if it builds up. So catalase swoops in to save the day, breaking down hydrogen peroxide into harmless water and oxygen.
It's like a tiny hazmat team neutralizing a dangerous chemical. And it does it super fast, too, right? Something about bubbles.
You're remembering right. Catalase is known for its speed.
Yeah.
In fact, one way scientists actually see this in action is with a bubble test. They add hydrogen peroxide to a sample, and if catalase is there, boom, bubbles. Those are the oxygen bubbles being produced. The more bubbles, the more active the enzyme is.
It's like a tiny little bubble party in a test tube, and the more bubbles, the better the party.
Yeah.
But besides speed, catalase is also incredibly efficient, right?
Absolutely. It's practically perfect in terms of its catalytic efficiency. We're talking millions of reactions per second.
See, now that's just showing off, faster than I can even think. Okay, we've talked about enzymes that build things, break things down, rearrange things. But what about the enzyme that powers all of that activity? The one that creates the cell's energy currency. That's ATP synthase, am I right?
Ah, yes. ATP synthase. This enzyme is a true marvel of nature, a tiny rotary motor embedded right in the membranes of our cells. It's constantly churning out ATP, which is the fuel for basically everything our cells do.
It's like the power plant of the cell, keeping the lights on and everything running. But how does it actually work? It's hard to imagine something so tiny actually rotating.
I know, it's mind-blowing, right? Imagine a water wheel, turning as the water flows over it. ATP synthase works in a similar way, but instead of water, it's powered by a flow of protons, those tiny positively charged particles. And this flow is created by something called the electron transport chain. It's like a difference in electrical pressure that drives the rotation of ATP synthase, and as it spins, it generates ATP.
Okay, so it's like a miniature hydroelectric dam using the flow of protons to generate energy. And to think all of this is happening at a scale we can't even see.
Right. And it's incredibly efficient, too. In fact, scientists were so fascinated by this tiny machine that they spent decades trying to understand it. They even won a Nobel Prize for their work.
Well-deserved, if you ask me. It takes some serious brain power to unravel those kinds of molecular mysteries. Speaking of molecular mysteries, what about those restriction enzymes we talked about earlier? They're like little molecular scissors, right? Why are they so important?
Great question. Restriction enzymes are a cornerstone of molecular biology research, especially for genetic engineering. Imagine you're trying to study a specific gene, but it's hidden within a massive strand of DNA. Restriction enzymes are like those precise scissors that let scientists cut DNA in exactly the right spot so they can isolate just the gene they want to study.
So it's like having a scalpel instead of a butter knife for DNA surgery, much more precise. But why is it so important to isolate specific genes in the first place?
For so many reasons. It lets scientists study what individual genes actually do. By taking a gene and putting it into a different organism, they can observe what happens. What proteins does it make? How does it change the organism? This has been a game changer for figuring out how genes work.
So it's like taking a single ingredient from a complicated recipe and seeing how it changes the final dish.
Exactly. And remember those sticky ends we talked about earlier? That's another cool thing about restriction enzymes. They don't just cut straight through. They leave these overhangs that are like molecular Velcro. Scientists can use them to paste different pieces of DNA together, even DNA from different organisms.
Wait a minute, are you saying scientists can basically cut and paste DNA like it's a document on a computer?
That's essentially what genetic engineering is. And those sticky ends created by those restriction enzymes are what make sure that the inserted DNA goes in the right place. It's all about precision at a molecular level.
It's mind-blowing. We can actually manipulate the building blocks of life.
Okay.
But okay, let's zoom out from the molecular level for a sec and get back to something a little closer to home digestion. We talked about amylase going to work on starches in our saliva, but I'm guessing there's a lot more going on in our gut than just that, right?
Oh, absolutely. Digestion is a whole symphony of breaking down food, both mechanically and chemically. It's a multi-step process, and enzymes are the star performers. Think about it. We eat this jumbled mix of carbohydrates, proteins, fats, and all of those big molecules have to be broken down into smaller, absorbable units before our bodies can actually use them.
It's like taking apart a giant Lego creation, breaking it down piece by piece so you can build something new with the individual bricks.
Perfect analogy. And just like you need different tools to take apart different Lego pieces, our bodies use different enzymes to break down different components of our food.
That makes sense. So besides amylase, who else is on the digestive dream team?
Well, once that partially digested food makes its way down to your stomach, pepsin joins the party. Pepsin loves the acidic environment of the stomach, and its claim to fame is breaking down proteins to smaller chains of amino acids called peptides.
Okay, so amylase tackles the carbs, pepsin handles the proteins. What about fats?
Ah, fats. We can't forget about lipases. These enzymes are made in your pancreas and released into your small intestine. They're the fat busters of the digestive system, breaking down those greasy triglycerides into fatty acids and glycerol.
Got it. And there are even more specific enzymes for different types of sugars, right? Like what about lactose, the sugar in milk? I know some people have trouble digesting that.
You're talking about lactose intolerance? That's where lactase comes in. That's the enzyme that breaks down lactose into simpler sugars, glucose and galactose, that our bodies can easily absorb.
So if someone doesn't make enough lactase, that's when they have trouble digesting lactose.
Exactly. They might feel bloated, gassy, and just generally uncomfortable after eating dairy. It's super common. Luckily, there are ways to manage it, like taking lactase supplements.
It's incredible how such a small enzyme can make such a big difference in how our bodies handle certain foods. It really highlights how crucial enzymes are not just for breaking down food, but for tons of processes that keep us going.
Absolutely. And we've only scratched the surface of the vast and intricate world of enzymes.
It really is incredible when you think about it, this whole microscopic world inside of us, these tiny enzymes working away, breaking down food, building new molecules, keeping everything running.
It's a beautifully orchestrated system for sure. And, we always talk about DNA as the blueprint of life. But without enzymes, that blueprint wouldn't be much good.
Right, like having a set of blueprints for a house, but no construction crew to actually build it.
Exactly. Enzymes are what bring that blueprint to life, translating those genetic instructions into actual tangible actions. They carry out these incredibly precise chemical reactions that make life possible.
And we've seen just how diverse those reactions can be. Building, breaking down, rearranging, even powering the cell. It's like a microscopic toolbox with a specialized instrument for every job imaginable.
And just like a skilled craftsperson knows exactly which tool to use for each task, our cells have developed these really intricate mechanisms to control enzyme activity, making sure the right enzyme is switched on at the right time in the right place.
It's like a perfectly choreographed dance, with every enzyme hitting its mark at just the right moment. But what happens when there's a misstep? We touched on enzyme deficiencies earlier. What are the consequences when those happen?
Well, when an enzyme is missing, or if it's not working correctly, it can really disrupt a lot of things. Imagine a cog in a complex machine breaking down. It can throw the whole system off.
That makes sense. So what happens then?
It really depends on the enzyme and what it's supposed to be doing. Sometimes you might see a buildup of a certain substance that the enzyme is supposed to break down. That can lead to all sorts of health problems. Other times, you might have a shortage of an essential molecule that the enzyme is supposed to help produce, which causes a whole different set of problems.
So it's like a domino effect, one disruption leading to another. Are there ways to address those kinds of deficiencies? Sometimes, yeah. Scientists have developed what they call enzyme replacement therapies. Basically, they supply the missing enzyme externally. Like people with cystic fibrosis often take pancreatic enzyme replacements to help them digest their food properly since their bodies don't make enough of those enzymes on their own.
It's amazing that we can actually intervene at that level, supplying the missing enzymes to get things back on track.
It really speaks to how far we've come in understanding these tiny but mighty molecules and how they impact our health.
And speaking of incredible scientific advancements, we can't wrap up without mentioning Cas9 and its revolutionary role in gene editing. It's like we've got the ability to rewrite the blueprint itself, right? Exactly. And that kind of power comes with some really profound implications, both scientifically and ethically. We're talking about potentially curing genetic diseases, engineering crops to be more resistant to pests, even modifying the traits of future generations.
It's like something out of science fiction, but it's becoming our reality.
It is, and that's why it's absolutely crucial that we approach these advancements thoughtfully, We need to have open and honest discussions about the ethics, the potential risks, all of it.
Absolutely. We have a responsibility to make sure these powerful tools are used for good, for the benefit of humanity.
100%. It's a shared responsibility, too. Scientists, ethicists, policymakers, the public, we all have a role to play as we navigate this new frontier.
It's exciting and a little bit daunting all at the same time.
It really is. But you know what? I'm incredibly optimistic about the future. I think if we use these tools wisely, we can unlock some incredible solutions to some of the world's biggest challenges. In healthcare, agriculture, sustainability, the possibilities are immense.
Now that's a vision worth striving for. Well, I have to say, this deep dive has been quite a journey. From the tiniest enzymes working diligently inside our cells to the global impact of enzymes like Rubisco, it's clear these molecular marvels are essential to life as we know it.
They absolutely are. And I think as we continue to explore their hidden world, we're bound to uncover even more fascinating secrets about these little machines. Secrets that could revolutionize everything from medicine to agriculture to how we understand life itself.
I can't wait to see what discoveries are on the horizon. But for now, we've reached the end of our enzymatic adventure. A huge thank you to you for bringing such amazing source material to the table and for joining me on this exploration of the incredible world of enzymes.
My pleasure. It's been a fascinating conversation.
And to everyone listening, the next time you take a bite of food or hear about some amazing new discovery in biotechnology, take a moment to appreciate the silent but powerful work of enzymes. They really are the unsung heroes of life itself. So until next time, keep those brains buzzing and never stop questioning. This has been The Deep Dive, signing off.